We need to earn our license to operate. We have to make a case to the public, a case to society that science is worth supporting. We can't simply hide away in our laboratories and do our experiments and just expect society to be comfortable with that. They have to understand what it is we're doing. They have to recognize that it's of some value to them. They have to be part of the discussion to know whether this sort of research is appropriate or not. What we must have is a public that's comfortable with science, not frightened of science, and who fully recognize all that science can bring to make their lives better. Biologists are very comfortable with details and particulars, but biology does have some great ideas, and I'm going to talk about 4 of these today and point in the direction of a 5th, which is rapidly gaining prominence, and one that I think will end up being in the same state as the original 4 ideas. The first idea is the cell. All life is composed of cells, so it's the basic unit of life, and the cell is the simplest unit exhibiting the characteristics of life. And so there's 2 parts to that concept. As is the case often in science, it was technology that begat discovery. And the technology was the invention of the microscope. About 1660, Robert Hooke was the first to use in a systematic way the microscope to examine living things. The most important observation for biology was that he took a razor and took a thin slice of a plant, a piece of cork, and within it he saw these little boxes. This was the first demonstration of the cell. Now, as well as technology often begetting discovery, another aspect of science is once one scientist has discovered something, everybody discovers it. Then single-celled organisms were discovered, and this was by Antony von Leeuwenhoek. One of the things he did was to make a scraping between his teeth and put it on a piece of glass and look at it under the microscope, and he was pretty surprised to see it full of microorganisms. And these are the first bacteria that were ever observed by Leeuwenhoek in about 1680, 1690. So this was the discovery, the realization that living things could be made of a single cell. Over the next 100 years, and we're talking from 1700 to about 1800, better microscopes, better ways to prepare tissue meant that the idea that cells not only made up plants, not only were seen as single-celled microorganisms, but also made up animals, became more and more prevalent. And by the beginning of the 19th century, the idea that the cell was the basic unit of life was born. A little later, under the very famous German scientist Rudolf Virchow, who invented essentially pathology, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. This is the statement that the cell is also the simplest entity that exhibits the characteristics of life. So that's my first idea, the cell. My second, the gene as the basis for heredity. From ancient times, it was observed that particular characteristics are passed down from generation to generation. So the question is, what is the basis of that heredity? Well, this was a problem in the 19th century. Plant breeders who were trying to improve crop plants by crossing different strains of crops. They were also thinking about the problem of how traits, characteristics were passed down. And it was complicated until Gregor Mendel came along. He argued that what you had to do was firstly accurately catalogue the types of plant forms that were being made, and secondly, to count them. But there were 3 particular reasons for his success. He actually experimented in a low-level way with about 6 different plant species. He discounted most of them because he couldn't make sense of the results. But there is one, which was the P, that he could make sense of it. The characters he was interested in were influenced by very few genes in the case of the P, whereas in the other plants there were many genes, and he would have never worked it out. So the lesson there is that you have to choose a system that you can make sense of. The second point, which I already have mentioned, is he recorded accurately what he saw and he counted it. And the third reason for his success is he generated a theory, a hypothesis to explain what he saw, and then went on to test it. And what he observed was that in the first generation after a cross, often one of the characteristics coming from one of those plants dominated. But if you then crossed that generation to itself, then in the second generation, the original parents both appeared again. And when he counted them, he got the famous 3-to-1 ratio that many of you may have learnt in school. Now, what this made Mendel think of was that there was something segregating here. And that led him to propose what we would today call essentially a particulate theory of heredity. He argued that in each plant there were two particles that were important for the particular characteristic that he was looking at, but only one of those was passed on to the gametes, to the pollen, for example. And the fourth thing he proposed was that one of those particles might dominate over the other in determining the characteristic. It was a brilliant piece of work, published in a decent journal, and nobody took any notice of it. Absolutely no notice at all. And then, 35 years later, at the beginning of the 20th century, 3 groups within a year did the same experiments, got the same results, and all of them rediscovered Mendel. Now, the first of them, I won't tell you which ones it was, tried to publish without referring to Mendel. The second group, who were a bit second, realized that it was better to acknowledge Mendel rather than acknowledge their competitors, so did acknowledge Mendel, and then they all had to accept that Mendel had scooped them 35 years previously. Now, why was it that it was taken seriously in 1902, 1903, and not in 1865? There were a couple of things that were discovered between, which I think might have been important. One was that the microscopists looking at cells had discovered chromosomes. The second observation was that Van Benedum looked at the chromosomes in a nematode. And again, he chose a material which only had 4 chromosomes. And when he looked at the gametes of this worm, they only had 2 chromosomes. And when you were making a gamete, you were reducing from 4 to 2, exactly as Mendel had predicted. So, the gene, that's my second idea. It's the basis of heredity. It explains genotype to phenotype, and it has still many, many implications. I mean, the sequencing of the human genome, for example, is an extremely important milestone. My third idea, evolution by natural selection. There's 2 ideas there, really. The first is that life evolves. And the second is that a major force for that evolution is natural selection. But actually the first of those ideas, that life evolves, was an idea that was prevalent for at least 100 years in quite serious ways before Charles Darwin. And I'm going to talk about one of them today, which was Erasmus Darwin. He was a doctor, a scientist, and a poet. And he was a great proponent of education for women, wrote a book about it, and set 2 of his daughters up, they were both illegitimate as it happens, 2 of his daughters up as teachers in the first public school for girls in England. He argued that life evolved, but it was his grandson, Charles, over here, rather more serious looking here, who gathered the vast amounts of information which supported the idea of evolution based on the fossil record and on his studies of living animals and plants. And what Charles did is he went on a boat trip for 5 years, collected lots of specimens, thought about what was going on, retreated to a country house near London, and wrote The Origin of Species, which he published in 1859. What he had here is he didn't know whether life had a single source or several sources, so you have A through to L, but what did happen is that all these branches and different animals and plants were going extinct. And what you see today is a consequence of the fact that all these processes have been going on. But every living thing up here is related by descent. What he also noticed was that these living organisms were often adapted to their lifestyles and environments. He produced his idea of natural selection, but I have to say even this idea is not strictly speaking only Charles's. It was co-discovered at the same time by another English naturalist called Alfred Wallace, wrote the idea up in 20 pages and wanted to know whether it was a good idea and sent it to Charles Darwin to ask his opinion. Charles Darwin, who had been working for 20 years on this idea, suddenly got this manuscript from Borneo explaining exactly his same idea. So what is this idea of natural selection? Is that within a species there are variants, differences, and these differences are due to inheritance. So there are inheritable differences we would now say were due to the genes. Secondly, those variants which are most successful will reproduce more, and so therefore pass on more of their genes within that population to the next generation than those that are not successful. Finally, these changes in genes will gradually lead to changes in the characteristics of the species, that is evolution. If you have the idea of the cell and the gene, it naturally will lead to evolution by natural selection. So this is an extremely powerful idea, and one that is really the most important idea in biology. It links everything together. The fourth idea, that is life as chemistry. Chemistry and physics, but I'm going to talk mainly about chemistry, is the mechanistic basis of life. It's the way you can understand life is through physics and through chemistry. Now, the replacement of Aristotle's idea of vitalism started really in the 18th century, and particularly in France. This is a painting of Lavoisier. It's by David. Now, what Lavoisier did is he began to do experiments on fermentation. He also noticed that when a guinea pig respired, breathed, then it was like slow-burning charcoal. He hadn't put it all properly together because the French Revolution came along. He was beheaded by the guillotine in 1794. It was France, so Pasteur picked it up 50 years later. And what Pasteur demonstrated was that living microbes were responsible for the chemical transformations that occurred in fermentation. That fermentation was a physiological act yielding chemical products necessary for the cell, and such chemical reactions are an expression of the life of the cell. And that whole idea developed into the biological science we now call biochemistry, and central to this is proteins, because the proteins are like little chemical machines or catalysts. I try to illustrate it here metaphorically, where the shape of the protein, the enzyme, changes the shape of the chemicals that we have going into it. When you take a cell, which is only 10 or 5 micrometers across, there are hundreds, if not thousands, of these chemical reactions going on simultaneously in the cell. All of these reactions are going on at the same time in a cell, in a tiny little space. The chemical environments under which these reactions occur have got to be in some way separated from each other for it to happen. So that when you think of the structure of the cell, the way I like to think about it is that you've compartmentalized all the chemistry into different places, so it can all occur at the same time. You have to communicate between these different chemistries to make sure the whole thing is properly coordinated. Now, this last idea is that you can understand biology in terms of informations and systems. Many biological phenomena can be described in terms of the interacting molecular components that make up a complex system. The best way to understand this biologically is in terms of information flow through the system. It's chemical systems for cells, neuron systems for brains, ecological systems for, in the environment. I'm going to give you 2 concrete examples so you can see what we're talking about. We can describe the double helix, as indeed it was originally, as the interaction between different molecules and atoms, how many angstroms the different atoms are apart, and you can describe the beautiful structure of the double helix. But that isn't the real reason why it's beautiful. The real reason it's beautiful is because actually what the double helix is, and that's the way one should view about it, is it's a digital information storage device. And when you understand it as an information storage device, it makes complete biological sense. My second example, is here. This is a governor or a regulator on a steam engine. And what happens is that it's attached to the spindle on a steam engine and it's rotating. As it rotates, these balls are thrown out by centrifugal force. That lifts a valve here which shuts off steam to the steam engine. That slows the engine down, and so the balls go back in again, which puts more steam into the engine. This is a classical negative feedback control. Taking circuits like this, you can combine them to make switches and timers and oscillators and toggles, which can produce complex circuits with information being fed through and being the basis of this complex system. An electronic circuit can be seen as a metaphor for all the connections going on in living cells. This is hardware, whereas in the living cell, the different components are connected through solution. It's wetware. Because you're connecting through diffusing molecules. And what that means is that you can very rapidly rewire the circuits through diffusible molecules, which is much more difficult with hardware. This is leading to the idea that the cell is a complex chemical system, and it acts as a sort of logical computational machine of linked different logical modules. So these are my 5 ideas. The cell, the gene, evolution by natural selection, life as chemistry, all of which I think would be well accepted. And the 5th idea, which I think others in this room would agree with, is that in the future, information and systems will be seen as key for biology. Thank you very much.